So thank you for inviting me to speak today. I have nothing to disclose. I'm going to be talking about the apolipoprotein or ApoE4 gene. So many of you will have heard of the ApoE gene. So we all carry a mixture of two copies of the three variants that are available. So the E2, the E3, and the E4 alleles. So the E3 allele, which is carried by about 75% of us, is the population norm, and it's relatively neutral in its influence on brain health. But the E4 variant, which about 17% of us carry, is the most well-established genetic risk factor for late onset Alzheimer's disease. So it's a really important target for preventative interventions, not only due to its prevalence, but also due to the kind of size of risk associated with carrying an E4 allele. So relative to the E3 population norm, if you carry one copy of this variant, you're about 4 times as likely to get Alzheimer's disease at some point in the future. And if you carry 2 copies, you're about 9 times as likely. So the effects of ApoE4 in healthy aging also make it a really important target for preventative interventions. However, the field is a lot more kind of murky as to how and when E4 starts to have a detrimental effect. So variability exists in the effects which are reported, both due to kind of the stage of the lifespan where we're looking, so what age our participants are, but also by cognitive domain and the type of assessment under study. So whether we're using standardized neuropsych, like the type we'd typically use in clinic, or whether we're using more experimental behavioral paradigms. So I've made this schematic just to kind of show you how I think the landscape looks. So at older ages, E4 carriers relatively consistently show disadvantages across a range of domains, including episodic memory, semantic memory, and executive function. However, curiously, in younger years, there have been reports, including from the group at Sussex, showing that E4 carriers show advantages. This isn't across all domains, but tends to be more executive and attentional processes. So my research focuses on mid-age as a point of strategic importance, where if we prioritize the use of sensitive cognitive tasks, we may first be able to identify early detrimental effects of ApoE4. And we can then use these cognitive markers to help test out interventions aimed at mitigating the harmful effects of this gene prophylactically. So how do we go about identifying cognitive markers of interest? So it's important to think about which brain areas are first affected by the progression of Alzheimer's pathology, and then work backwards from there, thinking about which cognitive processes these regions support. So I'm going to talk to you about tau. So tau is more closely kind of causally linked to the onset of dementia. And it first builds up in the medial temporal lobe. Specifically, this area of the medial perirhinal cortex is the region of first vulnerability. And this medial perirhinal area is involved in how we represent entities within the brain. So it sits at the apex of the visual processing stream, and it kind of manages how we take in these kind of visual features. So for example, the stripes on a tiger kind of binds them with our semantic memory. So we know that tigers have stripes and they live in a jungle in order to form a kind of cohesive representation. Along with support from the entorhinal and hippocampal regions in the medial temporal lobe, it's involved in how we discriminate between very similar representations. So for example, these two tigers, including how we discriminate between similar representations in memory. So we were interested in whether E4 carriers show differences in perceptual discrimination in midlife. So in a pre-registered study, with the help of NIHR bioresource, we recruited 401 individuals aged 45 to 65 years old. And we asked these volunteers to complete a 3-hour battery of purpose-built cognitive tasks and also self-report questionnaires from the comfort of their own home. Our sample was biased towards E4 carriers. So we had 183 E3, E3 controls, 172 E3, E4 heterozygotes, and 46 E4, E4 homozygote carriers in our population. And this allowed novel exploration, both of how being an E4 carrier in general affects perceptual discrimination, but it also allowed us to test our hypotheses that the disadvantageous effects of this gene would increase as a function of the number of E4 alleles carried. So we asked our volunteers to complete a Griebel's odd one out task. So participants saw an array of these three purple Griebel things, two of which are the same, but with slightly different rotations, and one of which is different. On the low ambiguity trials, relatively few perceptual features are shared between the two that are the same and the one that is different. So it should pop out, which is the odd one out. However, on the high level trials, so the high ambiguity, all three share a large proportion of visual features. So it places significantly higher demand on the perirhinal region in order to disambiguate them. So we use generalized linear mixed effect models to look at the probability of a correct response on this task. And we found that across low and high ambiguity conditions, there was a non-linear effect of APOE4 gene dose, in that the people who carried 2 copies of this high-risk variant across both the low and the high ambiguity trials showed poorer performance relative both to the control group and to the E3, E4 heterozygote group. So there does appear to be a disadvantage for the people who carry 2 copies of the gene. We also looked at response time for correct perceptual discrimination. Across the board, we found that our E4 carriers were slower. We also found this intriguing age by E4 interaction. So when we grouped our participants by 5-year age band, which you can see on both of these plots along the x-axis, with the number of E4 carriers, alleles somebody carries, there's an increase in the slope of age-related slowing. So in the younger age groups, the E3, E4 carriers and the E4, E4 kind of higher risk group tend to be quicker. But as they get older, they show a greater detriment in performance. So this suggests that our perceptual discrimination task is sensitive to disadvantages in E4 carriers by midlife. And this could perhaps be due to a gain in toxic tau pathology in this group. So we've identified a sensitive cognitive marker of interest. The second thing I want to talk about is whether we can use these cognitive markers to test out interventions against the gene in midlife. So specifically, we're looking at whether we can act on patterns of functional hyperactivity in E4 carriers in midlife and whether this influences their cognitive performance. So we've chosen to act on functional hyperactivity because in the kind of prodromal stages of Alzheimer's disease, we see patterns of increased fMRI bold response or brain activity in these memory-associated regions. And this appears to be consequential in driving the pathway of Alzheimer's disease-linked neurodegeneration. And what sparked our interest is that our APOE4 carriers also show patterns of functional overactivity in these medial temporal lobe regions from early adulthood. And this is both during tasks which typically recruit these regions and during tasks which don't typically recruit these regions. And so we're thinking it represents neural inefficiency in our high-risk group. So a group in the US drew on the link between epileptiform activity and cognitive impairment to test out a novel intervention against dementia. And that they found that a 2-week treatment with an existing anti-seizure medication normalized these patterns of hyperactivity in individuals with mild cognitive impairment, and that this downregulation of brain activity was associated with improved memory performance, specifically on our ability to discriminate these perceptually confusable images in our memory. So participants saw a sequence of images, and they had to say whether each image was new, like the first presentation of the Rubik's Cube, old, so an exact repeat of something earlier in the sequence, or similar to something they've previously seen, but not exactly the same. And it was on these trials they saw the benefit. So yeah, we wanted to test whether we could use the same intervention in our mid-age APOE4 carriers and whether it would affect their cognitive performance. So we've just finished collecting data for this study, so I won't report any results here, but I just wanted to show the design. So it's a double-blind, counterbalanced, placebo-controlled design where 60 middle-aged adults grouped by E4 status received both an active drug for 2 weeks and a placebo for 2 weeks under double-blind conditions. So we collected, after each drug phase, a whole collection of different kind of MRI scans and cognitive assessments, and also some blood-based biomarkers. We're hoping that when we're unblinded to genotype and drug condition, that on our task, which looks at our ability to discriminate between similar objects in memory, under placebo, in our perirhinal, entorhinal, and hippocampal regions, our E4 carriers will show elevated response. However, when they receive the drug, their brain activity will be brought more in line with the E3 control group. And that this reduction in activity will lead to improved performance on this task. So to conclude, establishing the cognitive consequences of carrying an E4 variant across the lifespan will progress strategies of early intervention. Repurposing an existing anti-epileptic drug is promising because it allows for more rapid translation from lab to healthcare. Future research is needed to consider how APOE may interact with other biological, health, and socio-demographic factors in midlife to really progress a more kind of precise approach to midlife prevention. So thank you for listening. So I just wanted to acknowledge all my collaborators. There's quite a few, mainly at Sussex and Brighton, but also at Johns Hopkins, and to thank the funders for this study, in particular Alzheimer's Society, ALUK, and the European Research Council.